in the soil or in potting media for a while, even without the presence of a host. But once we get enough rain, such as in 2016 and 2017, we had a lot of rain in the mild months of March and June, we see those spores start to germinate, and then they're able to find the roots of their host plants. Because remember, they have no natural resistance to this pathogen. It's brand new. And the terrifying thing is once that pathogen has actually made its way into the roots, it could care less about the environment. So I worked at the Nevada Department of Agriculture, and there's maybe 3 other states that are drier than Nevada. We had to do inspections for this particular pathogen because once it has made its way into the vascular tissue of that plant, it could care less what environment you put it in. It's protected in essence by that plant tissue. So this is the scary thing as we're going to move into a little bit. Moderate temperatures and moist conditions, aka the Pacific Northwest, are ideal conditions that we know so far regarding sudden oak death. Now this is a nursery situation. In the Northwest, we have a number of nursery plants, a number of nursery operations. I think we're about 11th in the globe as far as nursery production. The terrifying thing here then goes to those symptomatic plants, as well as those plants with early infections. So again, they don't care what part of the country you're going to ship them to. So the scary thing is people can plant seemingly healthy plants into their yard, and terrifyingly enough, these can then jump into other urban forests as well as native forests. So make sure people are choosing healthy nursery stock, as well as having some monitoring programs, checking to make sure your plants are still doing well. Another caveat of what we've seen with nurseries is this pathogen can survive in water. So if you've ever not been to a nursery system, you see that they water. Often they can recollect that irrigation water into an irrigation holding pond, and then they'll re-irrigate, potentially spreading the pathogen all the way through. So when we first started having the outbreaks of sudden oak death, we did a lot of irrigation pond monitoring to try and see if we were indeed finding that pathogen in the water supply. Now, unfortunately, because I work with plant problems, we see just symptoms. This is another Phytophthora cactorum, infecting a maple tree. Again, we see those rusty spots as well as the very severe discoloration in the stem tissue. And then again, I've mentioned that rhododendrons like to get brown spots all over their leaves. So this is why we actually have to go multiple steps further as far as detection. We have very similar and native species of Phytophthora that can attack similar plants, as well as a number of abiotic issues. The way I equate this is how many things can cause a headache, right? You could have had not drinking enough water, been in a weird light for too long, had a little too much fun the night before, or listening to me for 3 hours. I induce headache at 3 hours. So those are examples of symptoms that all can be caused by the same problem. So as far as diagnosis, the first thing that we tend to do is look for abiotic versus biotic symptoms. Can we confirm that this was salt stress versus an actual pathogen, and which pathogen? On our plants, we do tend to see fewer feeder roots. So that means those fine roots, they tend to be what are targeted by the particular pathogen. Those bleeding cankers, again, anytime you see a tree bleeding, you should probably get that checked out. That is usually related to pathogens as well as some insects. These are usually associated with rust spots, but do keep in mind that different species will have different responses. Again, leaf spots, if they are pathogen related, they will be associated with the tips and midveins, as well as you'll see discoloration on the other side of the leaves. Now, we do have commercial field kits, so if you're responsible for a large swath of land that has potential hosts and you're seeing some symptoms, you can get essentially, we call these dipsticks or immunostrips, but they function like a pregnancy test. You can take pieces of the affected plant tissue, grind it up in the little buffer solution, stick the stick in. 1 line means the test worked. 2 lines means that you do have a Phytophthora available. Now, if you find this, it needs to be immediately sent into a diagnostic clinic, or you need to contact WSDA for confirmation, because these Phytophthores are so similar to our natives and endemic species that we often have to go to DNA levels to actually confirm what it is. And I will tell you right now, you've probably gotten this over and over. We'd much rather have you send in that sample and be able to tell you, no, don't worry, it's not Phytophthora ramorum. So don't worry, send it in. It's better for us to know that it's not that species than let that sudden no-death escape into our native environments. So next I'm going to be talking about kind of a newer issue on our radar, Xylella fastidiosa. This is a bacterial disease. It's actually had many names throughout time, and it has many plant hosts. So first is Pierce's disease. Who likes wine? No one. That was very scary for a moment. So Pierce's disease was found in the early 1900s on wine grapes in California. Later, it was found on causing citrus variegated chlorosis, and then many of us know it more commonly as bacterial leaf scorch. So Xylella fastidiosa, its name actually holds quite a few clues. Xylella means it's xylem limited. This is the water transport system. I equate this to a tree is essentially made up of a bunch of straws sucking up soda. That's what the xylem kind of looks like. What happens is the bacteria grows in between these different sieves. Fastidious means we can't grow it. A lot of microbiologists rely on growing organisms in culture to identify. We couldn't identify this particular pathogen for a very long time. We'll talk about why that is in a second. But because we couldn't grow it, we couldn't see it, we didn't feel comfortable figuring out what it was. We also found out that there are 6 different strains. This is a very complicating factor because, as I talked about, it affects wine grapes, citrus, and many shade trees. So those are very drastically different plants that this can infect. What we have found that these strains can be exotic into different locations. One area might only have one strain, and it might be endemic or native, but it's when a second strain or a third strain comes in that things start to get a little weird, because honestly, they start recombining in the same plant. So if you were a microbiologist, this would have blown your mind. So this bacteria, again, keep in mind, this is a bacteria, but it's vector transmitted. So it relies on insects to move it from plant to plant. The insects specifically are sharpshooters and leafhoppers. Sharpshooters and leafhoppers, as well as spittlebugs. Now these two, and it's the family. We consider anything in that family possible to transmit this particular bacteria because it is so widespread. What is also troubling is these vectors are incredibly widespread. So these 2 families of bugs are present across the globe. So anytime you might have a plant introduction that has Xylella, you could potentially have a vector to spread it around. And as we found out in the cases of this is glassy wing sharpshooter and this is blue-green sharpshooter, the bugs themselves can be an invasive problem. So it can be kind of a double whammy or a 1-2 punch if these particular insect vectors come in and are still able to transmit this pathogen. So the symptoms for Xylella fastidiosa are marginal chlorosis or necrosis. That's what we're seeing about right here. So it's going around the edge. So what happens, I made the comment that the trees are like a bunch of straws sucking up a soda, and it starts to clog those straws. So it'd be like you drinking your soda with 5 straws, and if I start plugging it up, you're not gonna get nearly as much soda, right? So that's what's happening with these trees. So they're not replacing the water as fast as they need to. So they start burning on the edge. They start to look like they're drought stressed. And it's, again, the same symptoms, but a very drastically different cause. So scorch and this marginal chlorosis is the primary symptoms. We do see leaf wilt, drop, die back eventually, as well as death. How many of you have been hearing about the olive issues in Italy? We've been losing a lot of olives to this particular disease. So we have seen by the time we start seeing symptoms, we lose trees very, very quickly. Do keep in mind that these different species and strains of Xylella fastidiosa have different responses. In the citrus situation, we get damage to the fruit. So this is very terrifying for our citrus industry because it changes the size of the fruit as well as the flavor of the fruit. I don't like funky orange juice. I'm pretty sure y'all probably don't either. Asymptomatic plants exist as well. We have a number of typhoid Mary plants that allow the bacteria to reproduce but not actually cause any harm to the plant. One of the other key traits of Xylella fastidiosa versus, say, a true drought is we do see a sharp line, kind of a darker line, where we see the yellowing, chlorosis, and necrosis, and sometimes a nice classic bacterial pattern of a yellow border to the tissue. But again, I show this to remind everyone how similar this is to a lot of the drought stress. Again, from Nevada, I'm very well aware of drought stress. Y'all up in Washington might be a little less so. With the movement of the pathogen. So the insect vector, again, it can fly. Weather can sometimes move these organisms around or allow them to move further up in latitude. So we can see changes in their particular habitats. Humans are very, very good at moving species accidentally. But what we've found is that the movement of infected plant material has been the biggest issue regarding the spread of Xylella fastidiosa. Humans are what are driving the spread of this pathogen. So as we move around, nursery trade, plant trade has been moving this particular pathogen the most. And again, I made the comment that the two families of insect vectors are present across the globe. So we can accidentally move an infested plant and almost guaranteed that we will have an insect of the vector family present to do the dirty work for us. So if you would like to know more about Xylella, we will be having a webinar April 12th, 10:30 to 11:30. You're stuck with me again, unfortunately, so hopefully that's okay. But we'll go into much more depth regarding this pathogen. We'll get really, really nerdy and have some good fun with it. We'll also be talking about the basic of borer insects, so a little bit of the invasives, as well as just the general biology of it. So you can register, it's free on our website, and it will be recorded and posted later. As we've talked about before, if you see something, say something. If you're unsure of what to do or what you need, call me, call Jenny Glass. We're here to help you. You can reach out to your extension agents, use the Washington Invasive Reporting app, and we'll get these things taken care of. We're here to help because early detection is critical. The most important thing we have to keep these plants around and keep them healthy for longer. So same thing we just said right there. And if you have any questions, I'm happy to take them.
Time for a couple questions.
Yes. All right. Mm-hmm.
My understanding is that the sudden oak death hasn't been found in Quercus scariana yet. Is that true?
So far as I know, they haven't found it in that particular oak species as well. But USDA's APHIS would have the most up-to-date information. We do more studies, so I'd always be a little cautious just because maybe we haven't had that perfect combination of disease triangle to initiate infection. But at this time, I haven't heard of that as well.
So I saw that it affects some conifers. Is it putting our redwood forests at risk?
So the redwoods, I can't remember what I had just heard about redwoods. I do think sequoia can be infected by sudden oak death. I can double check that for you. But there is always a possibility. And again, because we don't know where it's from, we don't always have the perfect idea of what it's capable of.
So you haven't seen mortality from this in conifers yet, or?
You can see conifer death. It can be a threat.
Which ones? Just curious. Do you know?
That is a very long list. I'm happy to get you that information if you would like, though.
Okay, great. Thank you.
Doug firs, true firs are the ones that we know for sure. Okay. Oh, Sharon?
On the middle slide that you just had up, where you have the drought symptoms, with the drought on the edge, right, and only a partial edge. That's fairly commonly seen on rhododendrons, which makes me wonder now if I've been misdiagnosing, because it doesn't go all the way around.
Well, okay, so the comment is the fact that sometimes there's just slight margin or sections of the margin that start to show that necrosis is associated, usually with drought stress. So it is one of those. So far, we've done WSDA has done extensive sampling and we haven't had as big of an outbreak as we could have feared regarding Xylella. So at this time, it's just worthwhile checking. If you think it's going to be drought stress, really simple test: add some water. If it recovers, great. If your cultural management practices aren't improving the health of the plant, that might be time to start tapping into it. Because again, we have so many similar just worth reaching out to the clinics, WSDA, and your local extension office. We are going to help you get the accurate diagnosis. But yeah, I wrote it in. That's why you go very around very quickly.
Great. Well, thank you, Rachel. And